Today we will discuss a new paper where the main idea of that paper is that MLK elevation in the pancreatic cancer cells can reduce metastasis in the liver. So how they come up with this MLKL genes in the pancreatic cancer cells, that we will discuss in this figure. So what are the approaches that they have used is shown in the panel A. That is, they use this pancreatic cancer cells, organoid mice model, and external cohort validations. That is, surrounding the pancreatic cancer cells, how the tissues or cells behaves, or in other terms we can call them as tumor microenvironment cells. And the dry lab techniques, in terms of dry lab techniques that they have used, as the RNA-seq chromatography, this immunostaining, and single cell RNA-seq data. So they analyze the pancreatic cancer patients at T1M0 and T1M1 stage. And this difference between these two stages is that T1M0 has lower tumor size, less than 2 centimeter, whereas T1M1 has the tumor volume greater than 2 centimeters. And what they have found out, the dot represents the onset of pancreatic cancers, and the arrow represents the migration of the pancreatic cancers or metastasis of the pancreatic cancers in the liver, that can be seen here. You can refer to the actual figure to get a better idea. So from this onset, it is migrated to the liver. So first they did this. They used the clinical tissue samples of the pancreatic cancer cells, and they plot the heat map, and also find out what are the changes that are observed, genes that are changes or differentially regulated in the pancreatic cancer cells compared to the normal tissues, in the pancreatic cancer cells tissues compared to the normal tissues. And they did it for both this T1M0 differentially differentially expressed genes and T1M1 differentially expressed genes. And out of the differentially expressed gene in both the cases, the common genes that are differentially uh, expressed in both T1M0 and T1M1 are 216. And when they use these 216 genes to find out the pathways that are dysregulated, they find out that necrosis is the pathways that is highly dysregulated in both the using CAG pathway in both the in these 216 common genes. And this panel F represents the plots out of these 216 genes, how many genes are upregulated and how many genes are upregulated, that are shown in the red, and how many genes are downregulated, that are shown in the blue. And these are the CAG pathway. The G, it is against the CAG pathway to show what is the fold enrichment that has been observed. And it is found out that necrosis has the highest fold enrichment in the pancreatic cancer cells. Thereafter, they check the survival possibilities of the patients that have the high necrosis. So in this case, the OS represents the survival probability, and what they have found out is that in the patients that have the high necrosis or high this ML high MLKL genes has the lowest survival possibilities, that can be seen here. That the survival possibilities in the red color and the MLKL regions in the blue colors, that is the high MLKL will leads to the lowest survival possibilities, and the low MLKL will lead to the highest survival possibilities. This is the main gist of this figure. And after that, they check the actual patient samples about this MLKL presence. So in the case of this MLKL PDAC, they can see that there are some green colors are green fluorescence that indicates the MLKL presence. And in the case of this T1 M1, they can see that the signals for this MLKL are further increases, and simultaneously they also check the other conditions of the MLKL that is required during this upregulation of this MLKL, that is phosphorylation of this MLKL. And they can see that phosphorylations of the MLKL also is increased in the case of this pancreatic tumor cells, but not in the case of this cohort cells. So where this MLKL are high, so let me re-explain this panel I. So this first row represents where MLKL are low, and the second row represents where MLKL are high in the PDAC. That can be seen from the fluorescence signals of this MLKL, which is represented by green fluorescence, and the phosphorylated MLKL is represented by pink fluorescence. And this indicates that the MLKLs low are in the MLKL low are in the regions where MLKL low are in the regions where tumor volume is high, where MLKLs high in the regions where tumor volume is low, indicating that low tumor volume has a high probability. Low tumor volume has a high probability of spreading. Low tumor volume has a high probability of spreading across the metastasis, and that has been found out in their that has also been found out in the previously published literature, that low tumor volumes are spread. Uh, Probability of spreading of low tumor volume to the liver is high compared to the high tumor volume. And the one more thing that they have found out in the panel I is that the MLKL volumes are high in the case of high tumors, whereas it are low in the case of low tumors. So this could be the reasons that MLKLs could be the reasons that preventing the high tumors to spread across the different uh, organs or metastasis into the liver. And next they check the other proteins that are involved in triggering this MLKL, that is in the panel J. They did the Western blot to check whether those proteins are also upregulated, and they have find out that in the case of this T1M1, the upregulations of those proteins are high compared to the T1M0, indicating that this T1M1 has high this MLKL. So it will prevent MLKL has some properties to prevent this migration of this T1M1 pancreatic tumors to different parts of the organs, or prevent the metastasis of this pancreatic tumor. And these things they have also validated using the HNE staining or immunostaining of this MLKL genes in the T1M0 and T1M1. And what they have found out in the quantifications is that in the case of this T1M1, the relative amount of this relative amount of MLKLs are high in the case of T1M1 compared to the T1M0, when whereas the other amounts are also whereas the other amounts are not that much affected. That can be seen here. So this is the quantifications of the panel Western blots. So only this T1M1 only this MLKL genes are found to be upregulated, whereas other genes that are involved in this upregulation of this MLKL does not show any significant difference, except this phosphorylation of this MLKL that shows statistical difference. So from there they made a point that it's not the gene upregulations that might be occurring in the high tumors, or there is a change in the gene upregulations that might cause this high MLKL, as the other signaling pathways are at almost at same levels, except this PML phosphorylated MLKL. And however, the result shows from the immunostaining that when the MLKL is upregulated, then they can see that reduction when the MLKL is upregulated, then they can see the reduction in these tumors, both in the case of T1M0 and also in the case of this T1M1. So all this indicates that right now the MLKL elevations could reduce the metastasis of the pancreatic cancer cells into the liver at T1 stage. However, the reasons is not shown in this figure, how this MLKL works, but we will discuss those reasons in the next subsequent figures. So if you need to understand more about the analysis and the experiments in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.